So we've had our 5 minutes delay now, which we always allowed for Ian Howard to come along because it was always a little bit late. Unfortunately, we're going to have to wait a little bit longer now for him to come. But I'm very pleased that Tony's over here, Tony Howard, his wife. So that's very nice that you're able to come along to our Ian Howard Memorial Lecture. So for those of you who don't know Ian, did not know him, Ian was a great man, known for his very perceptive comments about theoretical aspects of psychology, and also his practical aspects. The university, the CVR, is full of pieces of equipment that he built and designed over the years that he was here as a professor. And one of the things I felt it necessary to do was to take our speaker here around and see some of these devices. Especially because when we were in the tumbling room, he was, which is a room that can rotate around the observer, he was very convinced that he was upside down in the way that many of the observers are. It's always a good experience to go into this room. This is one of the rooms that Ian made. But I had noticed in his CV that he had a paper there which was looking at the differences in brain activity between regular exposure and virtual reality stimuli. So we figured that probably this room, although it is of course a virtual reality room because we're moving around in a way that real rooms don't move, but this I think was enough to create a brain activity that was probably in fact comparable to real life. Nevertheless, I was a little bit worried because I do a lot of virtual reality stuff that there were these differences that were there. So to introduce our speaker today, this is Dr. Glyn Humphreys, who's the Watts Professor at the University of Oxford and also the chair in the psychology department there. He has many medals and recognitions, the Spearman Medal, the Prize for Cognitive Psychology, the Prize for the British Psychological Society, for example, and also seems to be for 4 professors at once. He's the professor at Oxford, but he also continues his honorary professorship at Birmingham, where he was before he came to Oxford, and he's also a professor at Leipzig and at Peking. So that seems odd to have 4 jobs. I heard they all come with salaries as well. So one of the things about our speaker today is his very broad set of interests, coming from psychology, attention, the results of brain injury, for example, and also on social cognition. So I think this is an excellent choice for the Ian Howard lecture because of the breadth that Ian, I think, brought and still remains in the sense of which is one of our strengths, the interdisciplinarity, which I think represents. So his talk today is called The Salient Self, How Self-Related Biases Permeate Perception.
Well, thank you very much for the generous introduction and also for inviting me here to give this special lecture. It's a pleasure to be visiting. The work I'm going to be talking about is a new branch, really, in our lab over the last 4 or so years, really, and it's very much conducted in conjunction, or this right way, with Jay Shui, who's a postdoc in the lab. So the work, when we set out, really, was thinking about how vision, how perception in general operates, and about arguments that have been around in the literature for many years, for instance, going back particularly to the early 80s, where people such as Jerry Fodor, David Marr, and so forth, argued that perceptual processes should be informationally encapsulated, that is, separated from much of the ongoing context that is around us, in order for perception to be veridical, for instance. More recently, though, there have been various other different views. For instance, the predictive coding idea, I think, is one example of that, which suggests that human perception, human vision, for instance, optimizes processing by being sensitive to the direct interest, the direct context of the individual in the physical and social environment. And indeed, having visited the tumbling room yesterday, I think it's a great example of how constraints about real-world knowledge, for instance, moderate our perception so that you actually feel yourself upside down rather than the world as the stimulus is changing. And what I'm going to do today is examine this kind of very general distinction between information encapsulated systems and systems that are influenced by our surrounding context in relation to work on a particular phenomenon, which is about how the personal social significance of stimuli seems to modulate our perception. The work starts out by looking at this particular effect, which is a self-bias effect. And there's considerable work really over the past 10 years or so showing that humans are quite strongly biased in our responses to information about ourselves or information relevant to ourselves compared to other people. And that's been demonstrated a lot in high-level processes. For instance, in memory, we tend to remember information that is directly relevant to ourselves compared with information relevant to other people, for instance. If we're asked to carry out things like trait evaluation judgments, then we show biases in which we're systematically more positive for traits when we link them to ourselves compared to when we link them to other people. And these effects also influence some, at least putatively more perceptual effects, such as face recognition. And so just to illustrate this, do people know who this character is? In England this would be instant recognition, but maybe not over here playing soccer. Well, exactly, it should be ice hockey. Okay, this is Wayne Rooney. He's a Man United footballer. And if Wayne was doing this experiment in which he would have to decide, is this face looking at me or is it oriented away? He would be quick to make that decision. He's looking at his own face. And he'd be quicker than responding to another face. This guy is called Steven Gerrard, who's another England footballer. Wayne would be quicker at responding to himself than to Steve here. So there are a lot of experiments demonstrating these basic effects. The question really is what may drive these effects? What's going on underneath it? What aspects of this self-related information are important? Is it just the familiarity, for instance, in some sense of ourselves compared to other people? What level of processing seems to be affected? Many of these tasks are quite high level, memory, even face perception involves various kinds of things. It could be on the decision side rather than the perception side, and so forth. And we don't know much really about these kinds of biases in relation to perhaps other underlying factors that may modulate perception. One such factor, for instance, is reward. It may be that information about ourselves is inherently quite rewarding, and it's reasons of reward that we seem to show enhanced processing to self-related information. So while there are a lot of experiments out there demonstrating self-biases, I think we don't know that much about exactly what's going on. And that was kind of our starting point for the work that I'm going to talk about today. And to start looking at this, we tried to develop really, almost embarrassingly simple paradigms to look at this. And the paradigm we developed involves setting up really quick associations between labels for stimuli and particular geometric shapes, for instance, or colors, or different kinds of stimuli. The idea was that in these procedures, because we're going to use what were neutral items, shapes, colors, and so forth, that things like the familiarity of the individual stimulus is going to play less important. And what I'm going to do is to talk about these effects, to look at questions such as how automatic are they? What are the brain structures involved in this? And how much does it, the self-biases relate to things like physical saliency? Are they very similar to effects of perceptual variables that we could manipulate? And so I'm going to talk about these effects. They'll all be based around this very simple association paradigm. And the talk is going to divide into 6 parts, which is quite a lot, so I hope you can bear with it. It divides into 2 parts, really. The first part about behavioral experiments. So I'm first of all going to introduce this very simple procedure and show how the effects are quite robust and they're stable across different individuals over time. I'm then going to look at the automaticity of the effect, where we vary things like the probability of occurrences and so forth. And I'm going to look a little about whether these effects of self-bias modulate our basal perception. Then in the second part, really, I'm going to talk about underlying neural changes that seem to go along with this. So I'm going to look at the underlying neural basis of the effect. I'm going to look at the relation to perceptual saliency, both behaviorally and neurally. And then finally, I'll touch upon the relations of these effects to perhaps other underlying factors, such as reward. So there's quite a bit of data that I'm going to go through, but I'll try and move it along at a reasonable pace and keep everybody with it. The bottom line conclusion, and I'm going to put up all conclusions in blue, is that this self-association can occur rapidly, and it seems to change what I'll call the social saliency of that stimulus. And this social saliency, perhaps a bit like perceptual saliency that we can vary in the stimulus itself, can modulate perceptual processing. So that's going to be my bottom line argument. So let's begin with the very basic phenomena. And here I'd like you all in the audience to be participants. You're going to be the subjects, and to get a feel of what it's like to carry out these simple studies. So what we started out by doing was just giving people a shape and a label and telling them, make an association between these two things. So for instance, make an association between the square and your best friend. Make an association between the triangle and a stranger. And make an association between you and a circle. So your best friend is a square, the stranger is a triangle, you are the circle. And you just tell people this, and then you show people stimuli that are either going to be the pairings that they've seen, in which case they respond with one button, or they're going to be repairings of those same stimuli. And then you press the other button. And it's as simple as that. So you're, the audience, you're going to say yes if it's the original pairing I just showed you, or no if it's a repairing. Okay, and prize is the fastest reaction time. Ready?
No. Yes. Yes. No. Yes.
Okay. So when people do that experiment, I don't know if you got that at all, but our question was obviously, is there a difference between these very simple shape and label combinations? And I wouldn't be here talking about this if there wasn't. But there is. What happens in the basic experiment is that these stimuli come up quite quickly. They come up for 100 milliseconds, they go off, and people are given 1 second to make the response. It's a response deadline. So people are being pushed quite hard as they're going through, making the response yes, no, etc. So they don't have too long to think about it. The data look like this, and over many years of running experiments, I've never run an experiment that works out quite as cleanly as this one did. Here you can see this is reaction time. The data are plotted in a slightly funny way. This is reaction time, this is accuracy. People are being pushed, so certainly initially they make quite a few errors. These are data for the stranger match stimuli, friend match stimuli, and these are responses to yourself. People are way, way faster and more accurate at making the match to that simple association relating to them. So it's a very strong advantage, and it happens after just a very small number of learning trials where they run through these associations. And that's the basic phenomenon I'm going to be talking about. One of the interesting things I think about the phenomenon is that if you take people into the lab, you test them on it, and so here are data where they're being tested time 1. This is the self, this is response to another stimulus like your friend, this is to a baseline or the stranger. You get that effect. You bring them back in a month later. This is the pattern performance. That's on RTs shown there. This is on D prime shown here. The advantage for the self compared to the other items. And if you look at individual differences in that, then you get quite stable patterns of performance in which the individuals who tend to show a very strong effect time 1 show the same strong effect on time 2. The individuals who don't show such a strong self-bias tend not to show it again on time 2. So there's aspects of this which is kind of stable across the individual. They show a trait-like bias in responding even in this very simple perceptual matching test. So, very simple test, trying to use neutral stimuli, which we can establish in this association test. It's robust and it's stable across individuals over time. And part of our thoughts in this was to take stimuli like this, but you might then plug them into other kinds of paradigms where we know something about what's going on and what's the neural basis for the effects, and then to see how the social variable starts to modulate these other paradigms. And that was our kind of approach. In part 2, I'm going to take a look again at the behavioral results and ask the question about how automatically do these effects occur. So you're set up, you're doing the matching. Can you stop yourself showing a massive bias towards the new stimulus? Can you facilitate the effects to stimuli associated to other people or not? So to look at this, we did something that's been done a lot in the literature, for instance, on things like the Stroop effect. Another example of perhaps a quite automatic bias that people show. Where in the Stroop effect, if you vary the probability of the incongruent trials, you reduce Stroop interference, for instance. So our idea here was to vary the probabilities of the different match trials that were occurring. And so in these cases, we've looked at things like this, where here would be a case where the self-match trials have a low probability, and you have 3 times the probability of the mother. In this case, we have mother rather than friend, and a stranger match trial occurring. Here would be a case where the self and the stranger have high probabilities, the mother's low, and here's a case where the stranger is low probability, and the mother and self are high. So subjects carry out the match test, and it's 3 times more probable that you'd get, for instance, stranger, I can't even remember what it was, stranger triangle, was it? Match rather than circle U, which is the one I remember. Okay, so that's the basic experiment, simple cognitive behavioral kind of study. What I'm going to show are reaction times here, differences. Well, it's actually the efficiency of performance, but it's differences relative to a case where the probabilities are all equal. So the probabilities are all equal in the first study I showed you. And the question is, if we play around with the probabilities, how do we shift performance? So if performance is plotted here in a positive way, what that means is that the self-bias, or no, the responses to the self get faster. So I hope that's okay. I'm plotting it relative to when they're all equal, and we see what happens when you vary probabilities. So the question is, what happens on low probability trials? So these are all the cases where the self is occurring with a low probability, when the mother stimulus is a low probability, and the stranger stimulus is a low probability, relative to when they're all equal. And what you can see is that for the self matches, there's really not much of a cost. It doesn't matter if this is a higher probability, or at least equal probable, or low probability, you're just as fast to the self. But what happens to the other stimuli is that there are costs. You get slowed in responding to these items when they have a low probability of occurrence. You can also look at what happens when you have higher probabilities of occurrence. And here there are different couplings of the stimuli that have higher probability. So in one case it's mother and stranger, and this is what happens to the mother and stranger conditions when they have a high probability. And basically they improve. So people get faster on these high probability trials. Both of them have a higher probability, and both of those conditions improve. The other conditions are whether self and one of the others, self and mother, or self and stranger, have higher probabilities. The data there look like this. So when there's a higher probability, the self gets faster again. So it does speed performance, shown here and here. But the other high probability stimulus, like the mother here, or the stranger here, gets no benefit. So what's quite interesting here is, I think, that when the self is with another high probability event, the gain is only for the self. When the self, when other high probability events are coupled together, they both gain. So that's varying probability. To summarize, on low probability trials, there are substantial costs for low frequency other items. You're slowed. If it's your friend, you become slow on that. If it's a stranger, you become slow on that. But there aren't really costs for the self when it becomes low probable. Secondly, when there's high probabilities, then if there are two high frequency other stimuli, they both benefit. But when the self is one of them, then there's no facilitation for the other high probability event. So that's a bit complicated, but I hope I can put it over reasonably. Our conclusions. One conclusion is, on these low probability trials, the self response remains fast, and the argument there would be that it's relatively automatic, the facilitation that you're seeing. The high probability trials, there are expectancies that modulate performance when one particular stimulus is occurring a lot. But if one of those stimuli is related to you, kind of unfortunately, that's your expectation. And if there's another high probability event, like your mother occurring, it doesn't win. Similarly for stranger. So our expectancies can be quite biased by these associations. So that's the basic effect, and that's looking at, can you prevent it by making it low probable, and if you expect it, what's the consequence for that? Thirdly, are there changes in perception? We're still doing a variety of experiments on this, but I'm going to talk about two particular effects, one of which has been a very simple manipulation that's been done in the cognitive literature a lot, which is to just reduce the contrast of the stimulus. A second one has been to look at the effects of redundancy, what happens when you present multiple exemplars of items, where there's arguments when you have redundant stimuli, you may get some integration in perception. So first of all, effects of contrast reduction. So in these experiments, we got people to make the associations, then we presented the stimuli either where the shape was high contrast, very clear, or the shape was in low contrast, and was more difficult to perceive. These are D-prime data for making responses to the self shape and responses to the shape associated to your friend. The point to note here is that for the friend, when it's low contrast, there's a drop in sensitivity. Okay, so that's the basic contrast effect. For the self, that's kind of protected against the drop in sensitivity. That you're less affected by that item. It's as if, even when it's low contrast, you give up the contrast to some degree. So there are effects on perceptual sensitivity, and one could argue from an interaction like this that there's some change in perceptual processing for the associated shape that's become linked to you in some way. So that's one simple kind of manipulation. The second thing that we've done is to look at the effects of redundancy. And I'll just talk about redundancy effects for a moment, if I may. There's a lot of experiments in the literature looking at what happens when people respond to a single item compared with when they respond to two exemplars of the same item. And what's often reported is that when you have two stimuli, for instance, if you have to identify if it's a circle or a square, you get two circles, then performance improves. You become faster at responding to the identity of that stimulus compared with when you only have a single exemplar. And that's called a redundancy gain, which is performance is enhanced when two targets are present relative to when only a single target is there. And that kind of gain may reflect, for instance, the fact that you're responding to some kind of integrated percept from the two items that are present. So we looked at that using these very simple association tasks. We got people to associate themselves or their friend with particular items. In this study, they had to associate two shapes with themselves, two shapes with their friend. And I'll say why in a minute. So here, you might have associated with you, you're a square and you're a circle. Either of those, they're both you. And then we presented on trials either a single item that people had to identify as that's them or that's their friend. Or we presented on the redundant trials two exemplars of the same stimulus. And that stimulus could be identical like that, or it could also be what I call conceptually related. For instance, the circle and the square are both you, so you are present on that trial, that's what you respond to. So that's the basic task. People get four shapes, two of them are associated to them, that's what you have to press the button to. Two are associated to their friend, they press the button to that one when the friend is present. They're seeing single items or they're seeing two items appearing. So, why did we do this experiment? Well, sorry, these are the basic data, first of all. So these are the basic data responding to the friend, single item friend, two identical friends, two who are both a friend but they're different shapes, and these are with the self. Basic point to note here is that for the self, in these redundant conditions, which are the two items one, you get a big gain compared to the gain that you get for the friend. So somehow having two of you is even better than having one of you when people are responding to that. But the reason we did these experiments is in looking at redundancy gains, you can carry out various more formal analyses of what's going on. So for instance, you can take formal models of the independent processing of the shapes on the trial, and you can look at whether the redundancy gain is bigger than you could get from the independent processing of the individual shapes. Okay, and so that becomes perhaps a more powerful analysis than just looking at mean RT differences with these stimuli. So one analysis of this goes back to Jeff Miller, which is called the Race Inequality, and I'll just explain it very briefly. What it's looking at is the probability of making a response at a given time to the stimuli. So this is the probability of making a response to a single item, for instance, an item in the left visual field. And this is the probability of making a response to a single item in the right visual field. And this would be the probability of making a response at the same time interval when you have two stimuli present, redundant. And the race inequality is a test of independent processing of these items, the two single items. And what it says is basically that in the redundant condition, performance, the probability of responding should never be better than the sum of the independent probabilities of responding to single items when they're present. And that's often looked at like this. So here we have data which is the cumulative probability of responding across time to these two different stimuli. I've tried to plot in red cases where that would be one single stimulus being presented, and this is a case where the other single stimulus is presented. The green dot is the sum of the probabilities for the two independent cases. This is a particular experiment where the redundant trials, which are shown here in the orange, are shifted to the left, which means that there's a higher probability of responding with redundant stimuli than the summed probability of single stimuli at a given time. And that's been taken to indicate that you have non-independent processing of the two stimuli when it's redundant. So that's a case that would be said to violate the race model, the independent race between two stimuli. And this would be a case where the race model would not be violated, because here the redundant, the summed probability is greater than the probability in the redundant condition, shown in the orange. So sorry to labour that a little bit. The take-home point is basically this is a more formal analysis of whether when you have two stimuli, you process them independently or you integrate them in some way. And what we try to do is apply this analysis and others to the case where these stimuli are associated to you or to your friend. So these are the data. You have these cumulative probability functions for responding. The top two are to the friend, and the bottom two are to the self. The thing to notice is this is the case where you have two identical shapes. This is the two shapes where they're not identical, but they both relate, for instance, to you, the circle and the square. This line here is the redundant condition, and the other black line is the summed probability of the single conditions. Redundant condition, summed probability. With the self, you have these shifts to the left when the stimuli are redundant. When the stimuli are related to the friend, this is the summed probability. You don't get any significant violations of this summed probability of independent processing of the items. So, with the redundant self only, the stimuli violate what seems to be independent processing of separate stimuli. And that's one reason for thinking perhaps that actually there's some response to the integrated percept of the two items, but only when they're related to you, or most strongly when they're related to you. So, the behavioural part of this talk on perceptual changes, the self interacts with stimulus contrast. It's not so affected by lowering the contrast, and it shows superadditive redundancy gains, which is at least consistent with enhanced perceptual integration. So those are sets of behavioural data to illustrate the robustness and the perhaps perceptual changes that seem to take place through this really simple manipulation. The second part, I'm going to talk about some neural changes that seem to also take place. And the first one is just a very straightforward fMRI study of people carrying out these matching tests. So people get the association, they then go into the scanner, and then they're matching. If it's you, circle, press yes. If it's you, triangle, press no. And then we look at which brain areas seem to be responding particularly to the you stimulus compared with the stimulus associated with other people. And these are data on that contrast. So this is a straightforward contrast between activity that's greater for the self relative to greater to the stranger associated items. And there are two regions that show an enhanced activation to the self-related items. And these are an area here, which is in the ventromedial prefrontal cortex, and an area here, which is in the posterior part of the superior temporal sulcus, shown here. And here are the beta values that you can see, the self being higher than the friend, higher than the stranger. Now, with fMRI, you have a lot of degrees of freedom to try and interpret what these brain areas are doing when they're associated with a change evoked by the stimulus. But the ventromedial prefrontal cortex is an area that has been reported a lot in other studies of self-representation. So when people think about themselves, when they make memory judgments about an item belonging to them compared to other items, then this is a region that's very typically activated. So there are reasons to think it's got something to do with responding to information about yourself or representing yourself in some way. The left posterior STS, again, has been linked to various things, but one proposal is that it's linked to what might be called more ventral parts of an attention-oriented network. And while that network's often been linked to the right area, the right homologue area, in aspects of social cognition, you can find activity in the left. And so our hypothesis is that what's going on here is that the brain is linking up self-representation to an attentional response to the shape that's become associated with the self, and that's sort of gains in saliency and produces enhanced activity when that shape is then presented. You can look at this in various other ways too. For instance, dynamic causal modeling is an attempt to look at changes in brain states or changes between different brain areas as a function of the variable being manipulated. And so here, this is a little model of these two regions, ventromedial PFC and the left STS. And changes in connectivity when you present self-stimuli compared with other stimuli. And essentially, there are correlations between the speed of responding to the self and the strength of this particular connection, or modeled connection, going back from the ventromedial prefrontal area to this STS area here. So the stronger the connectivity, the faster the response to the self. So stronger intrinsic connectivity, the more efficient performance when matching the self. And the idea being that somehow quick activation of a self-representation is then altering functional connectivity to bias responses to the associated shape. In contrast to that, if you do the opposite subtraction, so you look at which brain areas respond more to the stranger compared to the self, then you get regions shown here, which is in more dorsal areas of a frontal cortex, and also in posterior parietal regions shown here, which are more classically associated with an attentional controlled network. So activity in the classical attention control network is actually responding less to the self and more to the harder to associate stimuli, which is consistent with these, the stranger, for instance, being the more difficult task and the more attentionally demanding task. And one thing that we're quite interested in is whether there are opposite roles of these networks. That, for instance, we may have one network that is responding to the self, to self-interest, if you like, in the environment, and another network that is more to do with executive control and the task-based control of what you're supposed to be doing. And maybe those things can trade off against one another. We do a lot of work with patients with brain injury, and we're interested in whether, for instance, patients who might have damage here the executive network start showing very strong self-biases because this kind of network is unregulated or less well regulated. So self-matching is affected by a neural circuit that involves this VMPFC and the superior temporal sulcus. The strength of connections relate to the behavioral efficiency of responding to self, and that's distinct from the network that we classically associated with attention control. Okay, finally, I'm going to move on to the last two parts. I realise there's a lot of data here, but in part 5, I'm going to look a little bit at the relations between the social factor we try to manipulate and what are more clearly perceptual factors that you can change in stimuli. And I'm going to look at the relations between what I'm calling social saliency and perceptual saliency. And previously, work that's been carried out, work in our lab, looking at the effects of perceptual saliency, have used hierarchical stimuli. That is, local, global, compound shapes that have been used a lot over the years in cognitive psychology. So this is an example of a study where we manipulated perceptual saliency, the perceptual properties of the stimuli. So in this study by Meverak et al., we took compound stimuli, so big letters made of little letters, and we had them either so that you had high contrast items differing in colour, which tends to make the local items pop out, so we'll call that locally salient. And we have items that are all the same colour and they're blurred, which makes the global stimulus easy to see and the local stimulus more difficult. And we'll call that globally salient. And when you run standard experiments, where people have to discriminate, for instance here, is the local letter an S or an H, or the global letter an S or an H, you get different patterns of responses. So here is global salient, that's the blurred items, and responses are fast to the global in white and slow to the local in grey. Here's the local salient, that's these stimuli. Responses are now fast to the local target and slow to the global. Now that's no big deal, we've set the stimuli up to produce that. The point is that you can switch around a global or a local precedence by blurring or hiring contrast and so forth. But the important point to note, and so you as the audience is to know, is that by making these changes, you can produce a crossover interaction taking place here, in which whichever one's dominant depends on the salience of the item. Okay, so a second point about perceptual saliency is that fMRI studies have contrasted the case where the target is salient, item you're responding to, and the distractor salient. And when you do that, you find regions here in the left intraparietal sulcus, so in the posterior parietal cortex in the left hemisphere, that is more active when the target is low in saliency, so it's hard to see the target, and it's easy to see the distractor. And one argument is that this particular brain area, which may be part of an attentional control network, has to work harder when the distractor is really shouting at you. It's a very salient distractor, you have to ignore it for the task, and this brain area increases its activity. So there are two things, a behavioral marker, which is a crossover interaction, and a neural marker, which is activation here in the left IPX. That's produced by changing the perceptual properties of the stimuli. What I'm going to ask now in this part of the talk is, what happens if you don't change the perceptual properties of the stimuli at all? You just associate it with yourself or somebody else. So I'm going to report 2 experiments. The first one's a behavioral experiment. The second one is an fMRI experiment. And in both experiments, subjects initially have to form an association between a shape relating to themselves and a shape relating to their friend. And they then get these shapes put into the hierarchical figures, and they have to discriminate whatever the target is at the local or the global level. So the experiment kind of looks like this. People do 2 blocks of trials. In one trial block, they have to discriminate the self shape versus the stranger or the unfamiliar person. So in this self block, the hexagon would be the self, so that would be globally salient, and the circle is the unfamiliar person, that's locally not salient. Here, there's a hexagon at the local level, which is the self. So one trial block, self or stranger, self or stranger. Second trial block, it's friend or stranger that you're discriminating. Okay? Each trial you're cued is the target you're going for at the global or the local level, and then you respond. So these are data when you have stimuli that are incongruent, which are the most interesting trials. And these are the data with the self versus other discrimination. The point to note here is that this is a crossover interaction. If you're responding to the global stimulus, you're fast if the self is global, and you're slow if the self is local, the distractor. If you respond to the local stimulus, you're fast or faster if it's the self, and you're slowed if the self is the distractor. You have a crossover interaction here that qualitatively goes in the same direction as if you change the contrast or blurred the shapes. On the left is the case where it's not the self, but the friend versus the other person that you're discriminating. And here, it doesn't really matter very much which one is the target or which is the distractor. So I think there are effects of the social salience of these stimuli put into neutral compound letters, and the maximal for the self versus for the self relative to the friend. If you then have people running the scanner and do the same experiments, so the same basic task, but we're recording fMRI while people do it, then the results look like this. So here we have data from, in blue, where subjects are responding to the perceptual saliency and trying to ignore a highly salient distractor. In red are cases where people are responding to the social saliency, where you're ignoring yourself as the distractor. And yellow is a region that overlaps between the perceptual and the social saliency case. And if you look at beta weights within this region of activity that's shared, then you see this crossover pattern where activity increases where, for instance, the self is the distractor compared with when the self is the target. So the main point here is that we haven't changed the stimulus at all. We've just made people have an association with the stimulus. When you do that, behaviorally, it looks like the same effects you get when you change the stimulus. And in the brain, you also get quite similar areas being recruited to ignore a salient distractor. Our argument is that social significance operates similarly to the changes in the physical perceptual saliency, in behavior and also neurally. So if you've got a self shape at the global level, it's as if the stimulus is blurred a little bit. And if the self shape is at the local level, it's as if you turned up the contrast a little bit to that item. Okay, very finally, and I hope a slightly fun part of the talk, is about trying to look at the relations between some of these social things and maybe important underlying factors like reward. And as I mentioned before, if I could spell reward, that would be good. But does self-bias reflect intrinsic high reward value for things relating to ourselves? And that is an argument that's out there in the literature about why self-biases occur. Now, one of the things about this very simple paradigm is that it lends itself not just to looking at self-associations, but many associations. One of which could be reward. So if you run experiments like this, make an association between a value, £15 in this experiment, and a triangle, £5 and a square, and £1 and a circle. Then subjects carry out a match task where if they get £15 in a triangle, they should say yes. If it's £15 in the square, they should say no. We also give people a reward that reflects the reward value linked to the shape. So when you're responding correctly, yes, here, you get, for instance, 3 times the reward than you get here. Okay? So we try to make reward real in some sense, as well as just label associations. If you do that with these experiments, then you get quite similar effects to the effects you get with the self-association. So this is an experiment where we altered the contrast of the shape, similarly to that we've done with the self before. And this is the low reward stimulus, this is a high reward stimulus, and there's an interaction where contrast disrupts matching to the low reward stimulus more than to the high reward stimulus. So when you look at reward, you can get effects that quite mimic these effects of the self. But you can also start to do quite fun experiments once you start playing around with this. And one little experiment that we've done that I'll close on is to pit self-interest, if you like, or self-biases against reward. So in these experiments, we ask questions like, what happens if you make an association between a value and the label u? So you're £1, your friend is £5, and stranger is £15. So now you see stranger 15, you should respond yes to that. You should respond yes to you or £1, and you get, for instance, 15 times the reward on that trial compared to this trial here. Okay, all bets. What do people think will happen here? Well, who's going to win? You win. So these are defined values for this study here. This is for the self, this is for the friend, this is for the other. So our first result is that relative to the other, the high reward stimulus, the self continues to win. So the self-bias trumps reward values, at least up to the proportional reward values we vary. The slightly depressing result is the other contrast that you can see there, which is this one, which is that reward beats friendship. So in this case, people are faster responding to a high reward stimulus over their friend, even though normally the friend wins out in other circumstances. And here you get the decrease on the friend, even though that's got 5 times the reward value to you. So there's many, I'm sure many more imaginative things one can do with these simple paradigms to start looking at the trade-offs and relationships between things. But in this circumstance, there are interesting interactions, I think, between reward effects and self-bias effects. So within one set of bounds, self-bias overrules the effects of reward bias, although reward bias overrules familiarity, friendship effects. And this is one reason for thinking there may be some separation between the effects of reward and the effects of the self, and they may not be completely synonymous, they may not be completely the same mechanism. Or at least there are non-linearities in reward effects that are occurring in relation to the self. So, to summarize, I presented a whole sets of large amounts of data to you, but to try and put it together and to organize the results. The basic result is that after just a small number of trials, neutral shapes can be tagged by this social significance, and they show trait-like behavior, or people show trait-like behaviors. The bias seems to occur automatically, so even if it's a low-frequency event in the experiment, people are still fast. But if you expect yourself, you speed up to yourself, but then you don't expect other people. There's some evidence, I think, for some perceptual enhancement taking place in relation to those stimuli. Even in the brain, you can see changes, even after 12 trials, that reflect a particular circuit that's responding to self and self-oriented stimuli. And the changes match those you can produce by altering the perceptual properties of the stimuli. So you can at least mimic similar effects. And the self-bias effect does not seem to merely reflect differential familiarity or reward. So that's the kind of behavioral and neural studies. I hope it's of interest to see what you can do with really simple paradigms. Some overall points. Well, I think that techniques of this type may provide a way of trying to explore how social variables interact really with perception, with attention, and so forth, in perhaps a slightly more controlled way than is often the case. We would argue that self-bias modulates basic processing, and that perception is not, in that sense, informationally encapsulated. And in fact, we are open to social influences on perception. And maybe this is an important thing in human cognition, human survival, to produce enhanced attention and perception to self-relevant stimuli. For instance, if that baby is yours, you'd better pay pretty quick attention to it if you're there on the savannah and so forth, with it being present. So it's maybe not a crazy thing to have enhanced perception to things that are highly related to you. But maybe we can pick up on it in these very simple experiments. So, just remains to thank the various people who have been involved in funding this, and thank you for your attention.
Okay, so now we have some questions. Joe.
Yeah, it was a great talk. I love the idea of this association learning really quick. Have you, in your experience, found any patient groups that have problems in terms of developing this association quickly?
Oh, okay. I was just going to say, what a nice question, because look at the next slide. But this is not on learning it in the first place, I think. But someone down here, I hope I've got slides on patients. Okay. So, a lot of our work in our lab is neuropsychology, and we have a large tranche of patients with particular lesions coming in. And basically, you can do various kinds of studies, but big group studies involve testing every patient who's coming through the lab, and they get this little association test. And you can do various kinds of things. You can do a social association, like friend with square, a reward association. Patients really like this because they get rewards at the end of the experiment. And, for instance, an emotion association. So this person is the square, a happy person. So when you do that, you can get various kinds of effects. Maybe that's not the most. So here's one with, these are two patients who have come through the group of patients we've tested so far. This is the bias the patient shows. So this is patient 1 and patient 2. This is the relative bias to the self. So these patients show a big bias to themselves. If you look at reward, if you look at emotion, they don't show such an enhanced bias effect with them. You can get other patients who show other patterns. You were talking about the insula before. That, for instance, here patients whose lesions involve the left insula in particular. And these patients show a very strong emotion effect to the stimuli associated with emotion, but actually a reduced response to themselves. So they're not very biased to themselves. In fact, they're much less than them. So this is beginning days trying to look at the patients and what happens to selective damage to parts of these networks. But I think you can get selective damage and differential patterns according to the association. And this may be one way that we can try and pull apart, for instance, the self-bias from effects of reward, from effects of emotion, valence, and so forth. We've not done it kind of, there are obvious questions, what would happen with autistic individuals, or what happens in other disorders such as schizophrenia, depression, and we haven't gone there.
And this was one person?
This is actually two patients. So at the moment, the reason I haven't really reported this is we've run it on about 45 patients who have come through the lab at the moment. So these are selected patients who come out as abnormal in either showing a greater effect or a reduced effect.
Okay, and that's the average lesion down there?
So this is just over two patients, so you can't tell very much about the lesion. It's just lesion overlap over two patients. Yeah.
I'm going to ask the next question myself. So in Jaws, they have this boy that was attached to the shark. So at the beginning they had the scary shark and stuff, and then all the rest of the film they just had to show you this boy, and everybody sort of jumped out of their seats. And that's sort of the principle that you've got here. You've taken these shapes and you've associated them with words, and you're assuming that because you've associated the word self with a particular shape, that that's actually a valid association. It's not just that the word self is a more like easy to remember or more evocative word. I was wondering if you paired it with an Anglo-Saxon swear word or something like that to get some neutral words where you might have similar sorts of effects. So do you have any actual sort of independent justification that your use of the word self for all the results that you've shown actually do relate to the self itself, like actually real bits of your body or something like that?
Yes. Yeah. So first of all, it doesn't depend on things like frequency or whatever words. You can play around with those things. It doesn't make a difference. So the question is, can you get a handle on the self, and are the properties of the self you can maybe try and explore even this simple paradigm? I haven't got stimuli here, but we've done some experiments where you show kind of avatars. So, and you're seeing the avatars either from your perspective or from the opposite perspective. And the stimulus people respond to is just a colour that's presented on one of the avatars. And the self-bias effect increases when you see it from your own perspective compared to when you see it from a third-person perspective. So that's the first little tentative step along the way to try and see are the properties that are sort of special, if you like, kind of specific to the domain of the self that you wouldn't get with other kinds of stimuli. So I don't know what we would explore. I think it's interesting.
Hello, I'm wondering how you relate your results to the memory systems, where autobiographical memories, separate and can be quite independent from event or from procedural.
Yeah, I don't know actually at the moment. I think it's an interesting question about how some of these networks might link up. So in the imaging studies and stuff like this, we're not seeing classic memory errors being recruited, but people have formed the association and then responding to the perceptual stimulus. But clearly, I think there are quite robust effects in the memory literature about self-improving autobiographical responses and so forth, and episodic memory improving for self-related items. So I think there's definitely effects out there to start exploring the relationships and the interplay between these things. So that's all I can say at the moment. I think it will be very interesting to think of that framework and moving beyond, going higher up the system to memory as well as perception.
In the examples you gave, you circles and triangles and so forth. I think it's very hard to argue that those are neutral. I mean, circles have a rich connotation of perfection, wholeness. triangles, I believe it's been shown, are, or sharp angles, are more threatening. And so have you reversed these?
Yeah, sorry. In all of these experiments, there's counterbalancing, so it's not that the circle is always assigned to me. So the effects go over that. There may well be other kind of intrinsic things, you know, in the kind of natural association, but that's not what we're looking at.
So I was interested in the lateralization you get in the left hemisphere. And I'm wondering if, so in face perception, there's some lateralization, and there's also some hemifield effects. So you can get actually improved processing for stimulating the left hemifield. And presumably that's going to a right hemisphere, biased right hemisphere activation for face processing. So, and in your left hemisphere activation, you're making an argument that it's somehow equivalent to perceptual saliency, so it's kind of a perceptual activation. So I'm wondering if you would predict, or if you've tested, whether you have hemifield lateralization, a difference in left and right hemifield, or if you would predict such a difference.
So the answer is I don't know, really. And very often in these experiments, you just present the stimuli above and below to kind of, in a way, to get rid of some of those effects. A second point is, in all of the experiments I mentioned, the association is set up verbally. So you're making this association between you and the shape. So even when you present the shape afterwards, it's been established by itself. It's been established verbally. And so I don't really know at the moment whether some of the left hemisphere lateralization you see is because of that, rather than necessarily being left hemisphere. So I think we have to explore other ways of trying to set up the association, for instance.
Can you go back to your redundancy experiment?
Right.
I noticed that there didn't seem to be any self-advantage to the single item trials of that experiment. And just given how strong this effect is, I was curious about that.
No, well spotted. That's absolutely correct. It is. In the strongest effects that we get here are cases where you have the verbal label being used, for instance, in a direct matching case. So that's the strongest, and that's the original data I presented. One of the things that's quite difficult to avoid is when you're running, you know, and in redundancy experiments, you have to collect quite a lot of trials to do those more formal analyses, is to avoid people just setting up a stimulus response association between that shape up this one, this button, and that, which is all that people are basically doing. And in those circumstances, with a single shape, the effects are certainly weaker, I think, than in the case where you're doing the verbal matching to the shape. Although when you're getting double items, you do start to get stronger gains from both the items being present. So that is the case. That's my hypothesis about what's going on. And I think in these experiments, you do have to keep people tuned in to having the association as part of what they're using in the response set.
Just curious to get around some of these complications that Lawrence is raising, even James, if you get the verbal part, try and maybe do the association implicitly, like play a game where I have green squares and you have red circles. So I associate green squares with myself, and then do a test like you have, green squares and red circles. How do I work that?
I don't know. I'll write down that idea. So yeah, I don't know. We haven't done anything like that, but I think it would be very interesting to do. Yeah.
Why do you think you've got an extra speed self-advantage here? What does that increase in reaction time? How many useful?
Someone can speculate at the end, and I did try to speculate, which is that I think it may be important for you to spot your own thing. I don't remember, for instance, of your own tribe or your own child, for instance, in a certain circumstances. Work your mother. That's true. It does work well. Yeah, children are an interesting example actually, because mothers given this association with the child's name. can show bigger bias for the child's sake. So I think there are quite interesting things that you can play around with.
Okay, so two things. First of all, there's going to be reception in a few moments down in the behavioural sciences building, so we'll trace down there. And then what it remains to do is to thank you very much for your talk and to give you some CVR swag. There's a certificate here to that you've taken part in this important lecture, and CVR